Alright, hello. Our video is concerning the application of biochemistry and forensic science. We were tasked with the duty of critically reviewing 2 journals, and we decided to pick the ones based on DNA profiling. And as you can see on the screen, here's the general outline of what journals we picked. A bit of an introduction is that biochemistry is an important field in forensic science as it contributes greatly with things such as DNA profiling. There are both values and limitations in using biochemistry, which will be explained later on. There is also a bit on the screen about the development of the biochemical techniques. Concerning the second journal, a bit of the case history is shown on the screen. A 27-year-old woman filed a report at a police station saying that she was raped, and thus the investigation agency collected her undergarments and any genetic material that was found as to build a case for her.
So, I'll be explaining on DNA profiling. DNA profiling is known as DNA fingerprinting. So, there are few applications of DNA fingerprinting in forensic science. So, major application of DNA fingerprinting usually is in medical aspects. So the examples are paternity, maternity disputes, rape cases, murder cases, bomb blasts, killing by detectors, and war crime cases. So why is it DNA profiling is important? So the main goal of DNA profiling is it can assign a positive identification of the evidence that we found with those of the claimed suspect. So the claimed suspects are known as putative suspect. So this term is used in court for ages. So this is one of the basic requirements in the court. Actually, it won't prove the guilt, but it will throw more light on the evidence. So this what makes the evidence is more clear as we provide with DNA profiling. So the DNA fingerprinting usually patterns on the finger ball, and this has been used since ages. So the fingerprint refers to the maps of the peptide generated after electrophoresis and chromatography proteins of the individual. For basic fingerprinting, we have to know about the basic genome of the human, which is the genomic DNA. So these sources for the genomic DNA could be fresh or remaining. So the examples of the sources that we can take is from the flesh blood, dried blood stain, buccal cell, saliva, hair roots, urines, bones, pulp of teeth, semen, and vagina smears. So this technique actually allows us to study on various DNA from any sources that is obtained. We can know the variation of DNA. So the background of DNA profiling, it has 2 processes, which is the RFLP restriction fragment length polymorphism and the PCR-STR short tandem repeat.
RFLP, short for Restriction Fragment Length Polymorphism, is a molecular method of genetic analysis that allows individuals to be identified based on unique patterns of restriction enzyme cutting in specific region of DNA. It takes advantage of the polymorphism in individual people's genetic codes. The first step in RFLP is cutting the DNA. After the isolation of DNA, the isolated DNA digests the resistant enzyme, which recognize specific sequence in DNA and cut into various small fragments. These fragments have varying lengths depending upon the location of particular sequence on the DNA. This stage involves adding the resistant enzyme to the DNA using a pipette. After that, the DNA fragments were electrophoresed on agarose gel in order to separate the DNA fragments. Agarose gels is made from agar-agar, a type of seaweed that turns into gelatin when dissolved in boiling water. The negatively charged of DNA molecules migrate from cathode to anode. Shorter segments move faster and further away, while longer ones stay closer. The segments align in parallel rows. In the gel, there is a chemical called ethidium bromide, which sticks to all DNA fragments and allows the DNA to be seen when an ultraviolet light is shown on it.
Transferring DNA onto paper, the gel-separated DNA fragments are converted into single-stranded fragments by dunking the gel in weak acid. This helps the DNA lattice to be exposed rather than being in the middle of the double helix. The gel-separated DNA fragments are then transferred to white nitrocellulose paper, which now carries an exact replica of DNA on the gel, and this is called southern blotting. The southern blot equipment involves trays, paper towels, and a lot of solutions. Heavy books are placed on top of towels to squash everything down. The blot is then left overnight. Add in the radioactive probe. The probe is a small chunk of radioactive DNA of a particular sequence of letters that can determine which DNA fragments can be seen at the end of experiment. The probe then sticks to the fragments of the DNA that has the matching sequence. DNA fingerprinting. The probe is a sequence of 33 letters that is found in the repeated starters of the genome. The only DNA fragments that contain these repeated starters are seen at the end of the experiment as the dark bands. Setting up the X-ray film. Inside the dark room, the nitrocellulose paper containing the DNA fragments are placed against a piece of X-ray film in a large film cassette. The X-ray film is used to record the pattern of radioactivity on the paper where the probe has stuck. When the film is developed, it will have the pattern of bands on the DNA fragments where the probe had stuck. The film cassette is shut and left on the bench overnight or over the weekend, so that the film is exposed to the radioactivity for long enough to make an image. When the film is developed, it will have the pattern of bands on the DNA fragments where the probe had stuck. The film cassette is shut and left on the bench overnight or over the weekend, so that the film is exposed to the radioactivity for long enough to make an image.
PCR. PCR is known as polymerase chain reaction. Polymerase chain reaction is a technique that takes specific sequence of DNA of small amount and amplify the specific DNA. There is 3 steps of PCR, which is first denaturation, annealing, and extension. For these cases, there is a specific temperature for each step. What is a multiplex PCR? Multiplex PCR is the particular microsatellite or short tandem repeat that amplify, and this is one of the most useful method in molecular biology, which is used to compare specific loci on DNA from 2 or more samples. Why YSTRS is used for these cases? Because of the YSTRS is a short tandem repeats, and this is a focus on Y chromosome, which is male has X and Y, and the Y is for male. Next, for genealogical purpose, paternal lineage, and lastly, determination number of semen donor. The advantage of using YSTRS is saving in time and effort within the laboratory. Second, stable markers with very low mutation rates is ideal for forensic application, and this is very useful for forensic field. Next, can be used by tiny sample or degrade sample, which is damaged sample we can use also. Last, useful for small quantity of DNA due to PCR process. It also can for YSTRS. Good side, there is also has a bad side, which is disadvantage of the YSTRS. The first is increase the cost. Second is presence of multiple primary lead to cross hybridization with each other and the possibility of mispriming with other template.
DNA profiling is used widely in handling cases. There are strengths and limitations of the method applied during the DNA profiling. One of the methods used, DNA fingerprinting, can be used for individual identification in mass disasters or mass graves. For example, it can be used in identification of a person who has died more than 100 years ago. The next strength in DNA profiling is that the tendency to cut long DNA into small fragments precisely and in specific manners is higher with restriction enzyme. Next, revolutionized visualization of DNA fragments into molecular genetic analysis can be used extensively in blotting. Blotting is one of the methods in DNA profiling. Besides that, polymerase chain reaction (PCR) allows large-scale amplification in nose range of DNA in a very short time. This process can yield almost 1 million copies of the target DNA in about 3 hours. However, just like any other process of studying, most strengths come with limitation. For DNA fingerprinting, 2 samples upon DNA fingerprinting may match accidentally. This is because theoretically any DNA fingerprint only a very small segment of the DNA is analyzed and not the whole DNA of that individual. Next, the probability of chance matching of DNA fingerprints is reduced by different locus of locus-specific DNA probes. There are numbers of locus-specific DNA probes, SLP, MLP, or STR. An example for this limitation, MLP cannot be reliably used in DNA fingerprinting of the mixed sample like those of rape cases where multiple partners might be involved. In polymerase chain reaction, soils rich in humic acid and co-purified during DNA extraction process hampers PCR amplification by acting as inhibitors. It decreases the sensitivity during real-time PCR assays. In blotting, agarose gel that is used in blotting can be fragile many times, causing inability to use it for genetic analysis.
Conclusion: DNA profiling is one of the most important tools in the sexual assault, rape cases, murder cases, bomb blasts, paternity-maternity disputes, and killing by detectives. There are many methods can be used to detect the DNA in the sample taken at the site of crime happened, and each of method has their own advantage and disadvantage, which need to be chosen depends on the situation. To conclude, from the DNA fingerprinting in forensic and medical legal applications, they succeed to prove that DNA was matching between 2 samples, while for the DNA profiling of a rape case, they only get to matching the victim's DNA but not the suspect DNA, as they suspect the wrong person. From the result, they can catch the criminal identity. I hope that we can provide the sample as much as possible to detect the DNA in the sample and identify the DNA of the suspect to make it easier for court to catch. As the DNA fingerprinting only provide the proof of identity and not the proof of guilt, as was clearly observed in the famous trial of O.J. Simpson,